Hi everyone, my name is Atefeh Fard. I'm a postdoc at the Australian Institute for Bioengineering and Nanotechnology at the University of Queensland. Today I'll be talking about unlocking insight into cellular senescence through single-cell transcriptomics of aging mesenchymal stem cells. This project has different exciting avenues, but I'll be presenting only the data analysis or the bioinformatics part of the project. Aging phenotype is characterized by small specific subpopulation of senescent cells that secrete SASP factor, and these are called senescent-associated secretory phenotype, which contribute to aging. Cellular senescence is a state of cell cycle arrest as a cellular defense mechanism against unnecessary damage induced by different stresses, and these can include wound healing, cancer, and aging. Senescence phenotype is very heterogeneous and it's complex, and it's driven by a complex and dynamic nature through which several key molecular and cellular traits interact with one another. Using single-cell technology, we are able to study senescence population in a high resolution. We can understand the heterogeneity that is associated with senescent phenotype that wasn't possible before. So although many studies have identified different markers that are associated with aging and aging pathways, it's still unclear if there is a specific cell type driving the aging process, or if there are several senescent cell subpopulations that are either specific or universal to aging tissue. So here we use single-cell RNA sequencing of mesenchymal stem cells that are undergoing replicative senescence to understand the heterogeneity that is associated with the senescent phenotype. MSCs are multipotent cells. They have the ability to differentiate into different tissue types, including muscle cell, fat cell, and bone. And they have the ability to promote tissue repair by regulating regeneration and inflammation. And MSCs are very important for clinical applications, and there is an increasing evidence that MSCs are especially useful because of them being more feasible and much faster source for cell-based therapy in regards to stem cell transplant. So in this project, we are studying the heterogeneity associated with the senescence phenotype, but at the same time, we are getting some insight about MSC's aging and undergoing senescence. Okay, so here is the experimental design. So the main source of our cell populations were neural crest cells, and these cells were exposed to the MSC medium so that they transformed to mesenchymal stem cells. So we started with a very homogeneous cell population. We had 3 time points representing young, quiescent, and senescence MSCs, and 3 replicates per time point. We use chromium 10x single-cell RNA sequencing to capture single cells from all these 9 samples, and then we followed a standard single-cell transcriptomic pipeline to generate the count matrix and then perform the quality control, filtering raw quality cells, filtering lowly expressed genes, and looking at different metrics to check whether the quality of the data set is good or not. And then after that was done, then we performed clustering, we identified markers, we performed trajectory inference, differential expression analysis, and some custom analysis, including pathway overrepresentation analysis, cluster annotation, and looking at temporal differential distribution analysis. So this is the UMAP of all the cells, and they are color-coded based on replicate, time point, cell cycle phase, and cluster. So you can see that we have actually got rid of batch effect. There was actually a huge batch effect in one of the replicates. So we used Surrey integration pipeline to get rid of that batch effect. And then here, you can see they are color-coded based on time point. In the red, there is T0, and then up there T1, and then T2. And you can actually see the transition, which was really nice. For cell cycle assignment, you can see that most of the cells are actually at G1, which means that they are not in the proliferating phase. One of the most interesting clusters was this G2M, which means that the cells here are still in the process of proliferation. So you would expect to see these cells in T0, but interestingly, some of the T2 cells here are, some of the cells at this cluster are also T2. So here you can see the clusters. So we ended up with overall 8 clusters. However, cluster 0 here is very small, so we were mainly focused on the clusters that had higher cell population in. And so in this talk, I'll be mainly talking about how we went about to annotate these clusters, which happened to be really challenging. So again, what I wanted to check whether that G2M cluster, whether it contains any cells from T0 and T2. And then as you can see here is that it's actually these G2M cluster, there's some of our senescence cells that are proliferating. And that was really strange and interesting at the same time, because we knew that these cells are senescence, but it was interesting to see why they are expressing higher expression of, why there is high expression of G2M genes in these clusters. But again, majority of the cells were in G1 phase regardless of the time point. And then here there is a split of clusters based on time point. You can see that cluster 6 and 2, they are mainly comprised of cells at T0. And then at T1, you can see there's like cluster 5 and cluster 0. And then at T2, we have more clusters, cluster 1, 4, 0, and 5, and 3, which means that the heterogeneity in gene expression increases through senescence because we have more clusters at T2 as compared to the other time points. So what we did is that we divided or separated clusters based on early versus late time points. And here on the right, you can see the proportion of time points in each and every cluster. As I explained, cluster 1 and cluster 4, they are mainly T2 cells, which are senescence cells. Cluster 2 and 6, more than 95%, they are T0 cells, which you know that they are healthy young MSCs. And then cluster 0 and cluster 5, here they have, as you can see here, they have a mixture of T1 and T2, which means that they are in that transition process. Then we perform the analysis to identify the markers that are specific to each cluster. So we did that for time, and we did that for clusters as well. And as you can see here, T2 subcluster, T2 cells, they are more heterogeneous, as you can see in the expression as well. I'm just showing top 5 markers. So when we applied pathway overrepresentation analysis on these genes to check what they are or where they are enriched, we realized that so the pathways that were enriched, it was like very broad. We had cell cycle, ENT, epithelial to mesenchymal transition, proliferation, which was really broad, and we couldn't assign it to a specific cluster. But what I was really interested in is that where these genes sit, like what is their essentially function, what is their role. So what I did is that I took these genes and checked their gene family. I was mainly interested in the genes that were, I was mainly interested in the gene families that are important in senescence and aging, including cytokines and obviously SASP and cell differentiation markers. So cytokines, you can see that they are actually across the board in all clusters and time points. We can see them. Transcription factors, they are mainly in early time point clusters, which makes sense because transcription factors are responsible for regulation of gene expression. And once that proliferation is done and cells are entering in the senescence state, they are not proliferating anymore, and they are essentially stuck in that state. Again, it was interesting to see that there was no cell differentiation marker in early time point, but only in late time points. And it means that because these are MSCs, so they are trying to become stable by transdifferentiating into a stable phenotype like a cell type, but obviously there was no trigger in the medium, so they became senescent. And then SAS factor, you can see there is expression of SAS factors throughout the range in all time points and clusters, and that's because, well, obviously these are MSCs going through senescence. So, and SASP is one of the factors that is secreted by senescent cells. So this was also interesting to see that we can see that even in early time points as well. So I think the real story begins here, where we try to annotate these clusters, because obviously we were not expecting a lot of heterogeneity. They were all MSCs going through senescence, but at the same time we wanted to check whether there is or there are specific senescence subpopulations or senescence programs that are driving this process in aging MSCs. So we tried different approaches, automated one using packages like Garnet, SCCatch, single R. We couldn't get specific cluster annotation for these clusters. So we ended up using manual annotation using known markers. And then we also used the curated gene list that was given to us by our collaborator who are expert in aging and senescence. So in reality, in my perfect world, what I was expecting that each cluster shows a tendency towards a specific cell type. However, most of the cell type specific markers were highly expressed only in cluster 5. So really looking at cell type specific markers wasn't helpful because they were only expressed in one single cluster, and cluster 5 is the one that is like the transition cluster. So to characterize the cluster, obviously we use known markers that are specific to senescence, and we wanted to check are there any specific senescence programs in there. But the outcome was that the heterogeneity within MSC subpopulation going through senescence is not large enough for us to be able to assign a specific senescence program based on the known markers that are out there in the literature. We tried some lineage-specific markers to check whether there is a preference of specific lineage in different senescence subpopulations. But again, all markers were highly expressed only in cluster 5. So we ended up using the cluster-specific markers using pathway overrepresentation analysis. But as I said, the enriched pathways, the terms were quite broad. They were mainly cell cycle, aging, EMT, and they were not specific enough to characterize them. So we ended up investigating top 50 upregulated genes in each cluster, and we were able to annotate these markers. I am mindful of time, so I'll just go quickly through the cell type annotation process and also talk about one or two example genes that we chose for each cluster. So I'm starting from top cluster 5 here. As I said, they were a mixture of cells from all time point, and they were mainly expressing markers that cell type specific markers. So what we sort of concluded that these cells try to adapt and attempt to differentiate, right? They try to go towards stability, towards a differentiated state. However, there's no trigger, right? So they are induced by metabolic state and move towards a pre-senescent state that is cluster 0. Two examples of top highly upregulated genes in this cluster when NUPR1, which is known to convert stress signals into a program that helping cells to resist that the stress that is induced by changing the microenvironment. And there are NUPR1 is also participating in regulation of many processes, including cell cycle, apoptosis, and DNA repair, which totally makes sense in this scenario. And they also control cell cycle progression. The other interesting gene was VGN, which inhibits cell proliferation and is upregulated in MSCs during senescence. Cluster 1, SASP-related senescence, and these are the clusters that had high expression of SASP markers, GDF15, which is a SAS factor, and its increased expression is associated with cellular senescence. And THPS1, again, is also a SAS factor, and it's a protein that is produced by senescent cells. So again, this cluster we notated as SAS-related senescence. Cluster 4 is another T2 cluster which mainly contained senescent cells, and they were all mainly, all the oncogenes were actually upregulated in this cluster. G0S2, which is a tumor suppressor genes, TCL7, which is a MYC inhibitor, c-MYC is actually an oncogene as well, and S100A6 is also a MYC target. Cluster 3 is actually was one of the most interesting clusters because it had combination of T0 and T2 cells. It had combination of young MSCs and senescent MSCs. So we did twofold analysis. One was comparing within T2 cells. So we picked we subsetted these T2 cells and performed the analysis between the T2 and cluster 3 and the rest of the T2s. And the other way that we performed the analysis was essentially looking at cluster 3 and perform the analysis between T2 and T0. Overall, pathway representation analysis again was super general, mainly related to EMT, cell cycle, cell division. But what was really interesting in this cluster was that there were high expression of SASP markers like SERPINE2, but at the same time, there were high expression of genes that are regulating cell proliferation, which was really interesting. It means that these cells are somehow showing some features or characteristic of senescence, but at the same time, they have proliferation ability. Right now, we are actually in the process of validating these cells in the lab to check whether these senescent cells are able to proliferate further or not. So overall, cluster 6 and 2, they are healthy proliferative MSCs, and then what happens is that through time, they undergo metabolic stress, and then they reach their pre-senescent state, and then from there they'll have two sort of cell fates. One is the SAS-related senescence, one is oncogene-related senescence, and then these cluster here, we are calling them senescent escapees. These are T2 cells that have managed to escape senescence. With that, I'd like to thank member of my group here at AIBN, and they are all brilliant PhD students that gave me a lot of good feedback in terms of bioinformatics analysis. I also would like to thank our sequencing facility at UQ for performing the sequencing, and also our collaborator here at AIBN for performing the wet lab part of this project. I would also like to thank ISMB team for giving me the opportunity to present, and I'm happy to take questions now. Thank you.